We had here representatives of the European Commission showing the importance that microphysiological systems have at this moment and how they expect them to grow. And not only in terms of public funding, that they aim to reduce research with animals, but also pharma is very interested in this type of approaches because they can actually reach more reliable outcomes when they do this drug testing regarding toxicity or other types of outcomes they want to address from their drug development research. Hi, so my name is Sarela Garcia Santamarina. I'm a researcher in the human microbiome, interested in understanding human microbiome host interactions. I'm currently based in Portugal, but transitioning into Germany. And our interest in gut microbiota host interactions is what is bringing us here to the MPS World Summit to learn more about these systems and how can we implement them in our research. So this twinning project is a very nice project that we have now in place in Portugal. I'm coordinating it together with Sandra Tenreiro from the Nova Medical School. And so it's a project that is being conducted at two units of the Nova University, Nova Medical School and ITQB NOVA, to which I belong. And here what we are aiming is to gain capacity to be able to implement microphysiological systems to study more about models of disease in the Nova Medical School and more about host microbe interactions at the ITQB NOVA. So we felt there was this big need and this big interest in the community, but we didn't have the capacity. So this twinning project allows us to gain this capacity by establishing connections with 3 international partners. One from Jena, Alexander Mosik, a partner from Italy from the Human Technopole, Giovanni Faga, and 2 collaborators at the MDC Berlin, Agnieszka Rybak-Wulf and Stefan Kempa. Yeah. What we aim is to gain this capacity to implement microphysiological systems. So many of us are interested in organ-on-chip platforms, but other researchers are interested in implementing iPSCs and other stem cells-derived technologies. So we aim to first be able to collaborate more with external partners, also bring this capacity to the Nova University to train a lot of PhD students, postdocs, and also the PIs to be able to generate knowledge in this area. And we are already, not half through the project, a bit earlier, but we have already performed a lot of trainings for the students. We have given research money for 20 different groups to start to implement these projects. So I think it will be a very interesting path towards capacitation and bring Portugal to the European level in this type of research. Yeah. So we have, thanks to the partners, international partners that we have established many staff exchange programs. So students from ITQB NOVA and Nova Medical School can go to the laboratories of our external partners to learn the techniques there in hand. Through these staff exchanges. We have also training workshops in which also the international partners are capacitating our students, but more in a, let's say, not so personalized way, but more in a, let's say, with workshops that in which more participants can learn the techniques. And also we have the international partners sometimes coming to Portugal also to show us their hands-on techniques and ways to develop these models in situ, in the lab. So it was very significant for us being able to start the project, which was a bit complex due to the complexity of the project itself. But now what we are seeing is that people is already applying. Even we are maybe 8 months from the start of the project, but people is already in the lab establishing the techniques that have been learned during these 8 months. So this is very satisfactory, and I think it will be very promising also because the students that get the training will be able to train other students, so it's a very nice way. Yeah. So the international partners are obviously key to our capacitation. So we have, when we established the consortium, we wanted to make sure that we will have a diverse portfolio which could be incorporated into our daily research. So it is super important. Then we have established these 20 different small research projects because the funding is limited, obviously. But I think perhaps in 16 out of the 20 there is an international collaboration going on. So we have already established a network of collaboration. And I think this will bring more opportunities even for the future when we think about what can come next and what other types of networks we can establish together once this project finishes. So we have already sent people to our Italian partners where they have been differentiating human pluripotent stem cells. In the next few weeks, we will send some people to Jena, to the Alexander Mosig lab, where they will learn the gut-on-chip. So these are staff exchanges, so very personalized trainings. And I think next week, actually, some of our people will also go to the MDC to learn more about organoids and way to characterize organoids. And we have already conducted a workshop at ITQB NOVA, which was part of microscopy and how to handle samples, but the other part was organ-on-chip. So we were able to merge the microscopy course that we have already ongoing at ITQB NOVA with the organ-on-chip training. So it's yeah, very, very cool actually. Yeah, I mean, I think in this conference we already have at least 4 students that are supported by the MPS NOVA. We have other 2 students that are attending different conferences also related to the field. All those people, all the researchers that go to the training workshops and the staff exchanges are the young generation of scientists, so they are getting first-hand information of how to conduct this type of research. So I think, and they are already establishing their networks, their own networks, once they are traveling together. So I think, and they are very happy to have these opportunities, which otherwise wouldn't be possible. Yeah. We had here representatives of the European Commission showing the importance that microphysiological systems have at this moment and how they expect them to grow. And not only in terms of public funding, that they aim to reduce research with animals, but also pharma is very interested in this type of approaches because they can actually reach more reliable outcomes when they do this drug testing regarding toxicity or other types of outcomes they want to address from their drug development research. So I think in the broad scope this project is in the right moment and we are here to take the most of it and to bring it to the next level. Yeah. So what we would like to have or to build during these 3 years that we are actually conducting the project is to establish this strong networking, which we aim, when the project ends, to continue. We will have to look for other funding opportunities, but I think in pairways collaborations is already happening. So I know already, yeah, as I mentioned before, at least 16 people is already collaborating with international partners. So pairways is already getting into collaborations, but we hope and we expect that this network will be strong enough so that in the next level we can get more funding through other funding bodies to keep the network working together for longer time. Yeah.